Good morning, everyone, and especially thanks a lot for the introduction. So it's a bit overwhelming standing on the stage and talk on a topic. It's the first time for me. And I consider this as a very big opportunity and an honor to communicate science with you. So today's topic is divination by science, the art and science of gene editing. So as the name suggests, when you hear divination, you might correlate it with God, right? You know, something divine. So divination is basically, it's a skill or an act which can foresee future, or also it's something that can develop, something that will happen in future, which is basically everybody thinks that can be done by God, right? So on the course of this slide and this presentation, you will understand that divination can be done through science and only science. So before starting the presentation, as today is March 8th and it is celebrated as International Women's Day, so I'd like to acknowledge and give a tribute to all the women out there who have faced a lot of gender discrimination and also difficulties in life. Nevertheless, they have emerged successful in their life. And then I'd like to talk about few women in science. They are not so popular. You might have heard about Marie Curie. Even the video you saw about Marie Curie, very popular, right? But then there are many other women scientists who have given a lot of contribution to the scientific community, and then they have done so much for the science. For example, here there is Alice Ball. She's the first African-American woman to receive a degree from University of Hawaii. She has done a lot of work in leprosy. And then from Alice Ball to Yu, she's a Chinese chemist. She received her Nobel Prize recently in 2015 in physiology and medicine for developing a treatment against malaria that has saved a lot of people over here. So there are many more women who we have not heard of, but have had given their tremendous contribution to the scientific community. So now I'll begin my presentation about divination by science, the art and science of gene editing. So imagine a world where we can design our own babies. Like, I want my baby to have a certain skin complexion or a certain eye color. So you can design our own babies. So you might be wondering that, I'm saying, maybe I'm proposing a new script for a new sci-fi movie. But it is not like that. It is the near future. This will happen maybe in 20, 30 years, and it is still happening right now, and it has already happened. Okay, so one such news which shook the scientific community especially happened in 2018 when a Chinese scientist named he claimed that he developed the world's first gene-edited babies. So he claimed that he developed babies that are resistant to the HIV infection. So the babies were twins named as Lulu and Nana. And he also said that he did this using a technique, gene editing technique called as CRISPR. So what is CRISPR, right? So if you want to know about this technique and if you want to know about how this Chinese scientist did this amazing world's first gene editing babies, then you will have to first know about what gene editing is, right? So we'll take a step behind and then we'll talk about gene editing. So this gene editing, it looks like a very simple scientific term, you're editing something, right? But if you want to understand what gene editing is, then you have to first understand what a gene is, right? So what is a gene? Gene is nothing but it is an information that is carried down from your parents to their baby. So we basically inherit the genes, right? So our father gives us one set of genes, and the mother gives one set of genes, and then the baby will have two sets of genes. So these are just information. All the information how a baby develops will be in the genes. And then genes is made up of DNA. I think all of you would have heard about DNA. It's a building blocks of life. So our body, or in our earth, we have 4 basic DNAs. Adenine, thymine, guanine, and cytosine, ATGC. This is our genetic code. So just like I think everybody would be familiar about binary code, like 1, 0, of the computer language. Just like computer has a code, which is 1, 0, our body or our life also has a genetic code, which is ATGC. This gene or this genetic code, according to this, all the information about us is carried out. So this is our genetic code. So from a single-celled organism like bacteria to a multicellular, the world's largest mammal, which is blue mine, every living being has genes and this genetic sequence. But what makes us different is the difference in the genetic makeup. Every living being has a different genetic makeup that makes us different. So if you think about it in an evolutionary perspective, so every organism emerged from a single organism. So on the course of evolution, the species diverged, and then they finally are in a stage. Currently, all the species have been formed through evolution. So more diverse the species are, more difference in their genetic similarity. Okay? So for give an example, you say that humans are closely related to chimpanzees. So human and chimpanzees have 98% genome similarity. It's that 2% difference in their genome makes them different. So I've been talking about this genetic makeup and all that. So what exactly is that? You're talking about gene sequence, genetic makeup. So I'll give an example from a computer language or something like that. You might have seen in the video that's shown before also, when they was talking about computer code, they showed all these 1010. This is a computer code. In order to code this hello, my name is, the computer uses this code. This is the particular computer code. Similarly, our body also, we have a code too. That is our genetic code, this is how representation of our gene sequence take place. So the one DNA that I was talking about, the code, ATGC, all the different permutations and combinations of all that, it forms our gene sequence. You can see. So everybody has a different gene sequence. The order and the sequence will be different in every living being. Okay, that's what makes us different. Even in identical twins, the gene sequence is what differs. Okay, so this is our representation of the gene sequence, and all these genes will code to some physical or genetical trait in our body, in all the living beings. So let's see what a genes can encode or code to. So genes can code as simple as our eye color, our skin complexion. In case of plants, the seed, the shape and color of petals, everything is coded by some gene, okay? And it can also be as complex as our metabolism and also the human development. From the embryonic stage to a full-fledged human developed body, everything is decided or encoded by the genes that are inherited from our parents. Okay? Yes. So I told you that every living being has a separate gene sequence. There is a particular order also. So what happens if there is a mistake in that order, right? What happens when the genes go wrong? So when the genes go wrong, or the order in which the genes, which was supposed to be, if there is an alter in that order, you get genetic diseases, cancer, all this arises due to faulty genes. So for example, for a normal person, the gene sequence should have been something like that, the first two. But when there is a mutation is nothing but something that causes a change in gene sequence. You can see 3 yellow shapes over there. That is the mutated genes, okay? Due to this, a person can have a genetic disease. Genetic disease is nothing but that is carried down from your parents, which has been inherited, and it will carry down to generations and generations. And then you can also get cancer. So, for example, you have hemophilia and cystic fibrosis. All this are due to some faulty genes, and this has been carried down from our parents, okay? So, hemophilia is nothing but it is an inability of our bloods to form clots. So, once you bleed, it will never stop. It will keep on bleeding. And then you have cystic fibrosis. It is a mucus blocks the airways of our lungs. So, this will affect, there will be a problem in breathing, and that will affect. And then another important thing is the breast cancer genes. So I can say that the breast cancer genes, the BRCA1 and BRCA2 are the most popular genes. People have done so many experiments and research on this particular gene. And then scientists have found out that if there is a mutation in this particular genes, then you are more prone to get breast cancer. I'm not saying that 100% you will get breast cancer. If you have some mutation in this particular gene, then you're more prone to get breast cancer. So, and these can be inherited, can be passed down to your next generations as well. And also similarly in case of plants, you see some physical defects in plants. All this is due to some change in our gene sequence, some difference in the order of the gene sequence, okay? Now, think about some technology if we have, some technique if we have, so that I can bring back my mutated gene to my original order. So if I do that, the problem is solved. That exactly is what gene editing does. So gene editing, you know, it tries to bring back the gene to its original form. It will delete, it can insert, it can also modify the gene sequence so that it will bring back to the original form. So now think about the genetic enhancement that can happen due to this gene editing technique. Okay, so all these things are already being done, like for example, mosquitoes that don't carry malaria. So in mosquitoes, there is a particular gene because of which mosquito has the ability to carry this particular malaria infection. Okay, so we just have to remove now this particular gene from the mosquito so that the mosquitoes cannot carry the malaria infection anymore. That's exactly what gene editing does. And also about the mutations I talked about in breast cancer, if I delete that mutation from that breast cancer gene, then there is no problem. Yeah, the breast cancer gene are not mutated, so this gene editing can do it. And also apples that don't brown in the first top, you see apples, after when you cut it, after some time, the apples become brown. Now then people have developed a technology, basically genetically modified. They've engineered the apples such that after some time, the apples won't brown. So pet breeding is also, it's a fancy kind of, you know, genetic editing technology. You can use it for pet breeding as well. So gene editing is not something that came out of the blue. People have been doing it, you know, for a very, very long time, which is more prominent in the plants. You might have heard about HT beta cotton. Like these cottons are something which is resistant towards a particular herbicides. So all these have been done using a genetic engineering. You have modified the genes of the cotton in such a way that that can resist a particular pesticide or herbicide. Okay? So there are several methods to do this gene editing. So one of the popular method that the scientists use currently is known as CRISPR. It is a much faster, efficient, and cheaper method comparing to all other gene editing tools, okay? So I'll be talking about the CRISPR method as well today. So the CRISPR method, the final form of the CRISPR method that we see today is being proposed by Dr. Jennifer and Emmanuel. So, and out of a lot of scientists, there are 2 out of them who have developed this CRISPR technology. So I would like you guys to look at the quote that has been said by Dr. Jennifer. She said that we may be nearing the beginning of the end of genetic diseases. So on the course of this presentation, you will know why. Now let's see what a CRISPR and how CRISPR functions, right? So CRISPR is an abbreviation for clustered regularly interspaced palindromic repeats. Okay, it's fine. You don't have to know what exactly that means, and you don't have to get worried about the figure that is being here today, okay? So just like he's taking a video of mine, right? And this is going to get published in YouTube, but he might not publish the video in the same way. He will make certain edits in the video, and only then they'll publish it, right? So if I'm making some mistake in my video over here, presentation over here, further in future I can edit the video and then present it, right? Similarly, in CRISPR technique, we are also trying to do same thing. So we are taking the gene, we are trying to edit it. right? We are trying to cut it, and we are going to make our desirable editing in it, and then we are going to edit the gene. This is what we are going to try to do in CRISPR. So we have a gene sequence, okay? Now what we have to do is we have to find that target gene. We cannot go and cut it in any space, right? We can't do that. We need to first find the site where we want to make that specific cut. So that is being done using a guide RNA. So you have a guide RNA that will, as the name suggests, it will guide you to that particular position of the gene where we want to make that cut, okay? Now what we want to do is, we need to do that cut. In order to cut, we have a protein called as Cas9, which is in that picture, it is red in color. So that protein helps in cutting the gene sequence. Okay, so now we have reached a stage where we have cut the gene sequence. Now what? Now we need to join it back. Obviously, we need to join it back. So our cells in the body has an amazing capacity to do this. See, our body, our cells, has a mechanism known as DNA repair mechanism that will automatically, by natural, it is an inherent, innate mechanism in our body that will naturally join the genes back together. So, if our cells find that there is a cut in my DNA, my genes, so these cells will come and join it back, okay? So, but it is not an accurate process. It is an error-prone process. It might not join back the genes together. There can be a lot of errors in it, and that is a side effect of this process too. And you can also insert a gene using this process. You make a cut over here, and then if I want to insert a new gene, that is also possible using this CRISPR mechanism. So now you know how the CRISPR system works. How you know the gene editing works. Okay, you have a basic understanding of that now. Now we'll come into our first problems that we faced. That was the Chinese scientist editing, the world's developing a world's first gene editing babies, right? So how did he do it? So how would have he done it, no? So he said that he edited a gene called as CCR5 gene. So he edited that gene. So scientists have studied, and then they have understood that any mutation in this particular gene, CCR5 gene, makes our body resistant to HIV, okay? So there is this 1% population in Northern Europe. Over there, in their body, naturally, this particular gene is mutated. So for them, they are naturally resistant to the HIV infection. So scientists have studied about it. So the Chinese scientists also come to know about it. So he wanted to try this out actually in a human system. Okay? That's when he met a couple in which the father was HIV positive and mother was HIV negative. So they knew about the problems and difficulties you need to face when you are an HIV positive. And they didn't want this problem to happen to their child. So they voluntarily agreed to experiment on their embryo. So he took the embryo from this couple, and then he edited on this embryo, okay? So what he did was, he did the same method, the CRISPR method. He put that guide RNA, he put that Cas9 system, and he finally made that cut on this particular CCR5 gene, and he made that mutation over there. So he was successful in it. So he made that gene-edited embryo. And then using in vitro fertilization, he used that technique to implant the embryo into the mother, and then finally the twins were born. And then he's claiming that he has developed the world's first gene-edited babies who are resistant to HIV. So this is how the Chinese scientists went about with it, okay? So this news was not taken in a very positive manner with the scientific community, okay? He had to face a lot of criticism from the Chinese scientific community. They stated that there is an ethical concern for doing this type of editing on an embryo. Let's see what are the ethical concerns, okay, of doing this kind of an experiment. First thing, of course, it's safety. because we do not know what the long-term effects of this technology are. This is fairly a new technology that has been developed in 2012, so we do not know what are the side effects and also the long-term effects of this technology. So, take the case of this gene. So, scientists have studied and scientists are saying that this gene, if you mutate, our body will be resistant to HIV. But we are not 100% sure that this gene will only link towards this HIV only. This gene might have some other function in our body as well that we do not know of right now. Maybe later in the future, we might come to know about it. So that is what I'm saying you about the safety concerns and the long-term effects of this particular method. So it's only after a lot of clinical trials this can be assured that 100% usage of CRISPR is 100% successful. So this is one of the reasons. Then there is something called as genome editing research involving embryos. So one thing you have to know is that if you edit something in an embryo, whatever changes you make in an embryonic level stage, that changes can be inherited by your baby. That can be passed down to the generations and generations as well. Okay, so imagine you make some editing and that has a bad effect. So that is being carried down to the next generation, which has a very bad effect to the society. In contrast, if you edit one of the adult cell or something like that, that edition, whatever edition you made in the adult cell, that cannot be passed down to your next generation. But any edit you made into an embryonic level stage, that can be passed down. This is the reason why people are against embryonic level of editing of genes. Also, there are many religious constraints as well. So embryo is nothing but a fertilized egg. So it has a potential to form into a baby. So if you try editing the embryo, and then if something happens and the embryo dies off, it is basically like an abortion. So people as religious, according to the religious sentiments, people are not willing to do this genomic editing research involving embryos. Like in UK, Europe, it is banned. It is illegal to do genome editing research involving embryos. And then there is informed consent. That is nothing but you need to have the consent from the party which you are taking the embryo from, and you need to have voluntarily consent should be there. And it is pretty difficult to get that too. So what happened to the Chinese scientist who developed this gene-edited babies? So the price that he had to pay for developing the world's first gene-editing baby is that 3 years of life sentence. Chinese government also faced a lot of criticism, and the Chinese government was forced to give a sentence to the Chinese scientist who developed the world's first gene-edited babies. So these are the negatives. Now let's just focus on the future applications of this CRISPR. So as I already told you, it has the potential to cure cancer, genetic diseases, HIV, and also in agriculture you can form a lot of genetically modified plants which has the resistance that these plants can live in any condition. All these can be done using this CRISPR tool, using this gene editing. So I would like to tell an interesting application as well of transgenic animals. So transgenic animals are nothing but, you have dog. Dog has a particular sense of ability to sense of smell ability. It has this particular feature in them. Now I want my cat to have this particular trait in my cat of the dog. So if I want to do that, I just have to take this particular gene from the dog and put it into the cat, so that my cat will have all the characteristics of the cat plus this particular gene from the dog. This is basically what is known as transgenic animals. I'm saying it in a very loosely terms, but this is basically the idea of it. So you might have heard about this woolly mammoth, right? Everybody who have watched Ice Age movie will know what woolly mammoths are. And these are extinct. 4,000 years ago, these were living in our earth, but then these are now extinct. Now the scientists are trying to bring back these extinct woolly mammoth, not in their own form, but as a hybrid of elephant and woolly mammoth. So what they will do, they will take this certain genes from mammoth, woolly mammoth, from the certain genes of woolly mammoth and implant it or insert it into an elephant, okay? Then you will get a hybrid elephant mammoth, okay? They're calling it as mammophant or something like that. They're calling it as like that. So maybe in the near future, you know that woolly mammoth has a resistance to live in very cold places. So if I take that particular gene and put it in the elephant, now the elephant will have that particular trait, right? Now the elephant can withstand in this cold weather. Maybe, you know, in near future, you can see elephants will be running around in the Arctic region in ice cold conditions. So this is possible with gene editing using CRISPR technology. Now, these are all I'm telling about the future. This can happen, this might happen. I'm always using that future words. So, but now I'll give you certain examples in which presently this research is going on and people have already done a lot of research, gene editing, using CRISPR technology. So in a first, doctors in US use CRISPR tool to treat patient with genetic disorder. So this particular lady, she volunteered to do this experiment on herself. So she has this particular disease called a sickle cell disease, which is a genetic disease. So normally our red blood cells, RBC, it is in a round shape. But in case of a sickle cell disease, the RBC will be of a sickle cell shape. So obviously the sickle cell shaped RBC cannot do all the functions that a normal RBC does. So due to this, people face a lot of clotting and pain, a lot of adverse effects of sickle cell disease. So this is due to a particular mutation of the gene in our body. So what they did, the doctors, what they did was they edited it back. They used this gene editing technique and then they tried to edit back the sequence into a normal, just the way I've been talking about. And finally, she is showing prominent results. They are not saying that she's been 100% successfully recovered, but she is showing prominent results and improvement. And also another example I would like to share is this happened in 5th March recently, that they used this CRISPR treatment, they inserted directly into our human body. So in this case, what they did was, they took the bone marrow cells from the body and then they did the editing, all that in the lab, and then they transfused it back into our body. This was not a direct injection. But in this case, they inserted the tools of the CRISPR directly into her eyes, into the patient's eyes. So this is also a hereditary blindness disorder which caused blindness during childhood. So this is also because of a mutation caused in a particular gene sequence. So what they did, they again did the same technology. They edited back, they deleted that particular mutation. So they are also showing, so even this treatment is also showing prominent results. So these are the results which are happening right now. This is not the future, this has already happened. So maybe in a couple of years, this might be 100% recovery. We have to wait and see what happens in 2-3 years. You might get a 100% recovery status as well. So now I want you guys to come back to this quote which Dr. Jennifer had told, that she told that we may be nearing the beginning of the end of genetic diseases. Scientists don't blabber. Scientists only say stuff which has scientific reasoning and logical reasoning. So the results that you just saw over here are the evidences of her statement. She said we might be beginning for the end of a genetics diseases, and scientists and doctors are giving the proof of her statement. So I'd like to end my talk by saying that divination is something you can be done only through science, and there are a lot of evidences for it. We are giving proof. Let evidence lead, right? Yeah. So I'd like to conclude my talk. And then these are some of the references that I used. And I would like to thank all the organizers of Essence Bangalore, especially Srijit. He had contacted me and helped me along the presentation. I'd like to thank you all for listening to my presentation.
Neurons English is your one-stop free-thinking channel. Subscribe now and get instant alerts on videos from Essence Global.